right.
Good.
Yeah, I'm looking forward to your talk, Christian. Let's hear it.
Yes. Thanks, Dan, for giving me the opportunity to present here. Um, I'll present a project that we have been working on for the past 5 years. Try to understand how the bird retina gets its oxygen and nutrients because it lacks internal blood vessels that is at least part of the mammalian retina for providing nutrients and oxygen to these neurons. Um, it's a project that I did during my late postdoc years, and then together with 8 undergraduate students that has been extremely excellent, who has been in the lab, and then together with a range of collaborators that have been providing access to state-of-the-art facilities and equipment. Um, bird retinal metabolism is not a big field. So in case you think that I have misinterpreted some of the data and you think it could be viewed differently, then please let me know. And also, if you think that like new ideas might pop up, then also let me know. I'll be really happy to discuss. As you can also hear, my voice is losing out on me, so I really hope I can keep up for this hour. When we think about gas exchange, then what we're really considering is the final step of the oxygen transport cascade, where oxygen diffuses from the red blood cell into the tissues. We can use techniques to measure the distribution of oxygen from the capillary endothelium through the entire thickness of or through a tissue, where we will see that the oxygen levels tend to decline as the tissues take up more and more oxygen. And then at some point we reach a step where no more oxygen is present because the tissues have taken up all the oxygen. And for many neural tissues, this limit is around 100 micrometers. So that means that highly metabolically active tissues needs to have a dense network of blood vessels to ensure short diffusion distances such that all cells gets the oxygen and nutrients that they need in order to function. So if we look at a highly metabolically active neural tissue such as the brain, we see very, very dense networks of blood vessels that delivers those needed oxygen and nutrients to the brain. But one tissue is different, and that's the retina within the eye, as you all know, because it has this inverted design where all the neurons are packed in front of the photoreceptors. So if a very dense network of blood vessels are introduced into the retina, that will scatter and absorb the light, or at least there's the risk for it, before light reaches the photoreceptors. So there's a trade-off between like oxygen delivery to the retina and light transmittance in the retina. So for most animals, they only have a choroid in the back that delivers oxygen throughout the retina, where the same like diffusion limitations apply as in other tissues. So that means that there's probably a limit to how thick
you can't hear this, I hope.
Can you not hear?
Doug, we could hear you.
Okay, thank you.
I'll mute you.
Sorry.
Good. So if we consider the retina, the thickness of the retina as the diffusion distance for oxygen, then the typical limitations for gas exchange of around 100 or 150 micrometers apply very well to species that have poor vision. That could be a cave fish or a bat or an electrical fish that have a thin retina that is not packed with a lot of neurons, and where oxygen can easily diffuse from the choroid into the whole thickness of the retina. But let's consider some animals that have much sharper vision. That could be a falcon or a tuna. They have very densely packed photoreceptors, sometimes positioned in multiple layers. They generate multiple layers of synapses and interneurons, and that is connected in multiple layers of ganglion cells. And that markedly increases the thickness of the retina and thereby also the diffusion distance for oxygen. So those species that have very high acuity vision, they must also have solved some key physiological challenges with respect to gas exchange and metabolism in order to get the retinas to function with this altered retinal morphology. If we look into mammals, which you are all experts in, mammals have actually introduced blood vessels into the retina, but at a relatively low capillary density compared to other tissues that provides access to oxygen, but at the trade-off of potentially having some light scattering within the retina. We can measure the function of these blood vessels. We can measure if they actually provide oxygen. These are measurements done in Wei Li's lab, where we measured the oxygen levels throughout the retina in ground squirrels with similar techniques as Robert Linsenmeyer uses. So where we can insert an oxygen-sensitive glass electrode into the eye and measure in space where oxygen is present. And what we can see is that in the positions where we have blood vessels inside the retina, we get an increased oxygen level such that the retina is constantly kept oxygenated such that it can maintain its aerobic function through the entire thickness of the retina. This vascularization, it also allows for the retina to increase in thickness. Because now with the introduction of blood vessels inside the retina becomes less limited by oxygen delivery from the choroid, and that means that they can pack more neurons into the retina and expand the thickness of the retina. So we have compared the thickness of mammalian retinas that only have a choroid to species that have intraretinal blood vessels, where we can see that the thickness is almost 2 to 3 times longer in species that have vascularization, which suggests that vascularization is associated with the evolution of better vision in mammals. We have tried to look into this in a completely evolutionary context to see what kind of species of mammals have blood vessels inside the retina. Is it something that is just within a single group of mammals or distributed more widely? So we looked into all the classical literature and combined it with our own data, where we can see that in the current living species that are present here at time 0, some species are positioned or are listed in red. They have blood vessels in their retinas, and some species don't. We can use phylogenetic comparative methods to try to come up with information about when did vascularization originate, where we can see that it actually originated 2 times independently within the marsupials and 1 time in the crown group of placental mammals. And then it was secondarily lost multiple times within the placental mammals in species that are less reliant on vision. So that could be a rhino or a bat or some other species that are less reliant on vision. So this data on mammals suggests that oxygen supply is linked to the evolution of a higher visual acuity within mammals. So that oxygen and vision are linked in these animals.
Christian,
Yes.
I'm sorry, we never really sort of nailed this out beforehand, but are you okay being interrupted, even though I've already done it?
Yes, definitely.
Okay.
Yes.
And then, I mean, so are you suggesting that I'm not reliant on vision? Because I, at least in my daily life, feel incredibly reliant on it.
Yes.
So where does the argument sort of, how much reliance on vision as a proportion of sensation is this threshold, or is there a threshold?
At least we can see that mammals that are dependent on vision, that have good vision and high visual acuities, they also have vascularization. Including us, we have quite good vision for mammals. So whenever species have packed a lot of cells into the retina, that inevitably expands the thickness of the retina because it needs to accommodate a lot more synapses and interneurons. And that must be supported by additional blood vessels into the retina to support that metabolism and increase diffusion distances.
Okay. Sorry, I think I might have interrupted to ask the wrong question. So I'm gonna wait and let you continue.
Yes, but feel free to just interrupt in case you have any questions. Good. So based on these first findings, we then tried to look into the bird retina. The bird retina is quite different because it entirely lacks internal blood vessels compared to mammals, but the thickness of the retina is actually much larger. So in, for example, some species of eagle, the thickness of the retina is more than 600 micrometers. So that means that oxygen needs to diffuse more than a magnitude longer compared to our brains. The diffusion limit for the human retina is around 140 micrometers. So somehow these birds must be able to provide enough oxygen into the retina and get it to diffuse 4 times longer compared to a mammal. That's at least what we had in mind when we started off this project. The birds are highly metabolically active. They have a body temperature that is 42 degrees, so that means that they have elevated metabolism compared to mammals, which should also take up more oxygen and more energy. The idea in the literature on how the bird retina gets its oxygen, because people thought that it does, is that it is a structure within the... Sorry, say again? Okay. The idea in the literature was that it is a structure within the eyeball of birds called the pecten, which is like a vascular extension of the choroid that must be able to provide the additional oxygen that the retina needs and simply supply it from the vitreous side of the eyeball into the retina. But this has not been tested experimentally. So initially we wanted to test this idea, whether the pecten can even provide enough oxygen to keep the retina oxygenated. So again, we applied this technique of measuring the spatial distribution of oxygen in animals. We used zebra finches as a model for these experiments because they are similar size as a mouse. So we could use all our mouse equipment on the zebra finches. So we could anesthetize them, keep them physiologically stable, and then insert one of these oxygen-sensitive glass electrodes into the vitreous of the eye, and then gradually lower it through the entire eyeball, through the pecten and into the retina. Then we get like traces like this, where we have at distance 0, we have the choroid with very high oxygen levels. Oxygen levels drop as we go into the retina, and then they gradually increase a little bit as we go into the vitreous. We could apply some diffusion modeling to our data and calculate what is the flux rates of oxygen from the choroid and from the vitreous of the eye, which would provide us with the relative proportion of oxygen supply from the pecten and from the choroid. And when we did this, we could see that the pecten provides less than 1% of the oxygen that the retina receives. So the idea that what has been around in the literature for hundreds of years, that the pecten provides oxygen, we could falsify that idea based on direct physiological measurements. So that means that it is
the idea.
The idea that the pecten provides oxygen to the retina, that's the main function of the pecten. We could show based on this data that it does not provide much oxygen. It provides less than 1%.
Yeah. So I guess the question is that 1% enough to meet the energy demand? Oxygen consumption is related to energy demand. And so could it be that energy demand is just lower in that sort of on that side of the retina?
Yeah. It could potentially be, but it's at least within mammalian retinas, it's still a very active, metabolically active part of the retina. We try to then look deeper into the retina and see what are the consequences then since it's primarily the choroid that supplies oxygen to the retina. And the consequence is that half of the thickness of the retina is maintained in a state of complete anoxia. So there's no oxygen present in half of the thickness of the retina in the zebrafinches under stable physiological conditions. So this shows that the bird retina maintains an anoxic phenotype that is sustainable through the entire lifespan of the bird.
Christian, I have a question. So, when the bird's flying around,
Yes.
it's moving, right? And so the pectin could be moving a lot more when the bird is moving, and these were done under stationary conditions. Do you think that is it possible that can make a difference?
Yes, it could potentially do that, because then you could have convection inside of the retina, which will of course increase the flux. So yes, there is the possibility that if we were able to do the same measurements under conditions where the eye was moving, then that could provide like more oxygen to the retina. That's correct. And birds actually do, when they sit still, they do something called saccadic oscillations, where they just flip their eyes really quickly. And there's a preprint on bioRxiv at the moment that shows that it actually increases the flux of glucose from the pectin to the retina. So and it could be the same for oxygen.
Thanks. Yeah, that's pretty amazing.
Yeah.
Hey, Christian,
Yes.
has anybody tried to do like a vein occlusion of the pectin to see if preventing blood flow into it has any effect on the retinal anatomy?
There has been a study back in the 70s by Vincent Braque, who ablated the pectin, so did a surgery where he removed the entire pectin. And they saw some effects on pH within the eyeball, and they saw that the retinal neurons die off. So it means that it must have a function to support the pectin, which I'll also come back to in a few slides. But it would be a technique that could be really nice to get to learn, to be able to manipulate the pectin and then see what the consequences are. Yes? No, I thought there was another question.
I thought that might have been an echo somehow.
Okay, good. Then finally, we wanted to look into whether this is a diffusion limitation. So whether it is the retinal neurons that just takes up all the oxygen that they need, or whether they are limited by the partial pressure of oxygen that drives oxygen diffusion. So to test this, we manipulated with the choroid PO2. So we ventilated the animals on pure oxygen, which provides a very broad representation of choroidal PO2s. And then we measured under all those conditions whether the retina goes into anoxia. And here we could see that the oxygen levels needs to be above 150 millimeters of mercury in the arterial blood in order to avoid retinal anoxia. The maximum PO2 in a bird is similar to a mammal of around 100 millimeters of mercury. So this means that under no physiological circumstances that the animal experiences during its daily lives can it avoid retinal anoxia, suggesting that this is a chronic phenotype of these animals that they live with constantly throughout their lives. Good. This then begs the question, how does this work? We wanted to, we extended the in vivo measurements into more like cellular assays. And the question that we asked is, when we have an inner retina that is anoxic, what kind of processes are being activated here compared to the cells that are positioned just a few hundred micrometers further apart that are completely oxygenated? This was a new project, so we had to be kind of explorative in our approaches. So we wanted to measure whole transcriptome gene expression at a high spatial resolution. To do this, we used a technique called spatial transcriptomics. It's a technique where we can mount a retinal slide. We can do microscopy and staining to get an idea about the overall anatomy. And then we can permeabilize the tissue such that all the messenger RNAs that are within the tissue diffuses onto the microscope slide. And on the microscope slides are printed some capture molecules that can bind to the mRNA molecule so that they're captured directly onto the microscope slide. Each of those capture molecules have a spatial nucleotide sequence that is unique to that position on the microscope slide in which they were printed. And that means that we incorporate a spatial, you could say, GPS tag into each mRNA molecule, so we know where it came from in tissue space. We can cleave off the molecules, send them for sequencing, and then we get maps of gene expression for all the genes that are being expressed in this tissue, where we can see where they're distributed in tissue space. So for these zebra finches, we got around 5,000 different genes, where we could see where they're distributed at 5 or at a 55 micrometer resolution within the tissue. So when we link that to our in vivo data, because we know what is the oxygen level with respect to the choroid, we can actually get an intricate data set of in vivo PO2, whole transcriptome gene expression that we map to tissue morphology. So then we started to ask questions about what are the pathways for energy production within these retinal neurons. So we wanted to compare this to the in vivo measured oxygen levels. And then initially we took like the major pathways for energy production. And then within each of the gene expression spots, we calculated what is the overrepresentation of all the genes associated with, for example, glycolysis compared to the rest of the transcriptome in that gene expression spot. So we could see where a specific pathway is overrepresented in tissue space. So if we look at glycolysis, we can see that glycolysis is highly upregulated in the inner layers of the retina, which was the areas that were anoxic, but not really upregulated in the outer part of the retina that is oxygenated. The reverse pattern is for oxidative phosphorylation and beta oxidation, which are oxygen-sensitive processes that are primarily found in the outer retina and not in the anoxic inner retina. So this provided some initial ideas that the inner retina probably runs on anaerobic glycolysis. But anaerobic glycolysis is, of course, a good way to produce energy. You get fast rates of ATP production, but it comes with other physiological challenges. Because a retinal neuron that runs on glycolysis, that needs to take up more glucose in order to generate the same amount of ATP. But that glucose also needs to diffuse across extended distances because there's no blood vessel to provide that glucose. And then those neurons, they also produce lactic acid that would acidify the microenvironment in which these neurons are positioned and potentially reduce the function of these neurons. So to first understand if glucose can even get into the retina, we set up an experiment where we injected animals with radiolabeled glucose in awake animals, and then harvested tissues after 1 hour, where we could then measure the tissue uptake of glucose within different compartments of the body. So where we compared the eye to the brain, and where we would predict that if these retinal neurons runs on anaerobic metabolism, then they must take up additional glucose compared to an even glucose-hungry brain. So when we measured the glucose uptake in the same sections, we can see that the retina indeed takes up glucose, and it actually does that at a 250% higher rate compared to the brain. So this shows that the retina can indeed get the glucose that they need, despite that there's no internal blood vessels to supply that glucose. This then begs the question of where does that glucose come from? So typically glucose should cross some blood retinal or blood-brain barrier to supply that glucose from the bloodstream into the neurons. So we try to go back to our spatial transcriptomic data set and look for blood-brain barriers that had high expression of glucose transporters. And when we did that, when we mapped the GLUT1 transporter onto the histological sections, we could see that they were extremely highly expressed in the pectin. So this structure that was previously thought to provide oxygen to the retina, which we projected, we can now show that it has a representation of a glucose gland within the vitreous of the eye. We looked into a range of different glucose transporters, and we could see that it is only the GLUT1 transporter that is present within the pectin, suggesting that it could be that transporter that facilitates the diffusion of glucose from the blood into the vitreous of the eye. If there's a diffusion across this tissue, then there must also be a diffusion gradient. So in order to nail whether there's actually diffusion going on, we measured the plasma and vitreous concentrations of glucose, where we can see that indeed there is a gradient of glucose across this epithelium, strongly suggesting that glucose does move and does diffuse across the pectin. So it suggests that the pectin supplies that glucose that the retina needs. It is being fed from the inside of the eyeball via or from the blood via the pectin into the vitreous of the eye that then feeds the retinal neurons. Next question is, of course, lactate. How does lactic acid how is that removed from those retinal neurons? Again, we looked for different lactic acid transporters, for example, the MCT1, and similar to glucose, that is highly expressed within the pectin. It is the only transporter of the MCTs that are being expressed within the pectin. And we see extreme but reverse gradients of lactic acid from the vitreous of the eye to the plasma, suggesting that lactic acid is being dumped by the retinal neurons into the vitreous of the eye, that is then being taken up into the blood via the pectin through MCT1. Then finally we looked into...
Christian?
Yes?
Did you consider, your vitreous typically has a high concentration of lactate in humans as well, and a significant concentration of bicarb,
Yes.
right, to kind of obviously neutralize that. I don't know what the flow dynamics are in the bird retina, but if you considered also it being removed via other mechanisms than just the pectin, whether it be through your anterior chamber and out your TM, things like that, right? Other ways that lactate could be removed rather than back into the pectin.
Yes, that's right. We didn't then, but then we would but please challenge me here. But then we would probably expect glucose and lactic acid transporters being present other places within the eyeball, right? But we only see them in the pectin. But you're right; it could be like in the ciliary body where there could also be some transporters that could take up this. But you're right, potentially we actually only measure what happens in the vitreous as a whole and not specifically with where in the vitreous these processes occur.
I have a sort of another question. So,
Yep.
Based on my standards of the mammalian retina, which I'm learning is a very different system, GLUT1 and SLC16A1 are very highly, MCT1 and 3, I guess, are expressed on the RPE very strongly. Did you have enough spatial resolution really to get RPE, or is this kind of just like you're mixing outer segment and RPE and diluting that signal? Or do you have a sense of if you were to take a pectin and homogenize it, and RPE from the bird and homogenize it, is there similar levels of any of these transporters?
That I really wanted to test. We did. I'll show you in a few slides that we did single cell transcriptomics of the retina, but when we processed the data, then we couldn't find neither the RPE or the pectin endothelial cells. They were lost in the process, so we only got information on the retinal neurons and glial cells.
Okay.
But it's something that we would really love to do, like maybe do single nuclei sequencing where we are sure we will get those cell types and see what are the differences between an RPE cell and a pectin cell.
Yeah.
Is it similar cell types, or is it something completely different, or what is it?
Oh, the idea of the pectin as like the RPE of the inner retina is very appealing.
Yes.
But so I have one sort of other question. The diffusion coefficients for oxygen are much higher than that of glucose or lactate. So just from a purely like diffusional perspective, it would make much more sense, and this doesn't mean that it's actually what's happening, but it would make a lot of sense for oxygen to diffuse still out of the pectin.
Yeah.
Because, yeah, like your oxygen delivery from the pectin would still be greater than your glucose delivery from the pectin could ever be, just like your oxygen delivery from the choroid could be greater than your glucose delivery from the choroid.
Yeah.
So, yeah, can you make this make sense to me? I'm not really asking a question, so I don't know how it's easy to have a response.
No, it's totally fine. I actually thought that glucose had a higher diffusion capacity because like it's a more bulky molecule compared to oxygen, but the solubility is also very, very high, which increases the diffusion capacity proportionally.
Well, I think solubility and diffusion, they're related in getting to some downstream tissue. So maybe it is true that like that's important, but just the diffusion constant, once it's in solution, should be higher for oxygen than glucose. But is oxygen soaking into membranes better than glucose is? For sure.
Yeah, that's right.
Okay, if I can comment on that, I think if you're just looking at glucose and oxygen in I don't know, in water or so, you're probably right, Daniel. But here you're looking at a tissue which has for glucose specific transporters that are very, very efficient for transporting glucose into the tissue. And I think that may change the picture. It's not diffusion for glucose. For oxygen, I think it is. But for glucose, it's not really diffusion. It's more active transport.
A transporter can't let a molecule cross a membrane unless it's diffused to that membrane. I guess I have a sort of mental issue with the idea that a transporter can be faster than like the natural diffusion of the molecule through that substance.
I would love to see the evidence for this actually. Like Gamaliel actually made the claim that glucose diffused further than oxygen in some of his paper as well. And so and that was the claim that for tissues that were poorly vascularized, that maybe glucose may reach further than oxygen. So this is an argument. We couldn't find very good evidence for this actually in the papers that he referenced. But if any of you have that would be interesting. But that's why I'm actually buying the argument that Christian is making regarding the glucose. But I was wondering, Christian, if you could comment on the possible discrepancy in the 2DG labeling that you showed us previously. That seemed to be mostly in the outer retina and less in the pectin and the inner retina. Like, did it actually match your observation with the, or is it okay? No, maybe I was wrong. But your pectin does not seem to light up quite as much. It's just accumulates in the neurons, I guess, and it remains stuck there.
You need hexokinase.
Yeah, it's because it's being phosphorylated, but it doesn't move further into glycolysis, so it's being trapped in neurons that take up glucose.
So it's actively secreted by the pectin, and then it remains stuck in those neurons of the inner retina. Sorry, okay, I missed you. Okay.
Yeah. But maybe
Christian,
just
Christian, this is Jason Miller. Can you just to finish the thread that Tom and Dan had said, and you may have shown this picture, but is the pectin considered just a sort of a tangle of endothelial cells, or is there some other sort of set of cells there? You know, ciliary body is sort of double layered epithelium. What's the actual histologic structure and cell types besides the endothelium in the pectin?
It is primarily endothelial cells in a bilayered structure. There's also some pigmental cells, but other than that, I'm not sure what's actually there. But it's something that we would really love to look into, which we tried previously, but where we didn't get the data. But we would love to do a single cell transcriptomics characterization of the entire pectin to see the specific cell type distributions and also how they organize in tissue space. So little is known about it, but it's primarily endothelial cells.
I think you're safe to move forward.
Good. Sounds good. Yes, so one person had a question about like lactate levels and bicarbonate levels. We also looked into like what is the acid-base status of the vitreous of the eye, because there's obviously a lot of lactic acid being dumped into the vitreous of the eye. So what is the pH status? Also, what are the CO2 values? We could see that carbonic anhydrase 4, which is a carbonic anhydrase isoform that is present in our lungs, are highly expressed within the pectin, which could suggest that it has a role in CO2 removal. We could also see that the CO2 levels were around 75 millimeters of mercury. We've measured that both using direct measurements of CO2 with a CO2 electrode, and we have also measured it by sampling vitreous of the eye. So indeed, CO2 levels are extremely high, so the vitreous is under a massive respiratory acidosis.
Okay. Sorry. You can get. I'll ask a question when you're done with the slide.
Okay, good. Recently, a master student in my group is measuring the acid-base properties of the vitreous of the eye, as well as the plasma, where we can see that the vitreous values have very high bicarbonate compared to blood plasma, and it almost compensates the entire pH disturbance, meaning that they have normal pH within the vitreous despite that the CO2 levels are high and that the lactate levels are extremely high. So currently we're looking into whether the pectin or some other epithelium within the eyeball has acid-base transporters that could facilitate this pH regulation.
I absolutely love this data. So since you said you had a CO2 sensitive electrode, I have to ask, have you done any sort of penetration of that electrode through the depth of the retina to get the similar profiles as you've gotten for oxygen?
We didn't. So we worked together with a sensor lab that had just developed a CO2 electrode for doing air measurements of CO2. And we tried to make a sensor that was less sensitive such that we could use them under physiological conditions. And they made a whole range of sensors for us. And one of them worked. And that's the one that we used for those 4 or 5 animals. We only dared to just measure vitreous levels in one position because we were not sure about when to stop the profile through the retina, because we were not sure what would be like a marker of us being in the choroid and having to stop the profile.
Wouldn't arterial CO2 values be similar to choroid CO2 values? Or
yes, it probably would. But we were just very scared of doing those measurements with our single electrode that worked. So that's why we only did point measurements. But it's something that could be done, and which
I hope someday that you do it. I would be very excited to see what that looks like.
Yes, definitely. Yeah. Yes, so it shows that there's indeed a pH disturbance, but it's almost fully compensated within the retina or within the vitreous of the eye. So that means with this data, we can kind of rewrite the function of the pectin from previously thought being an oxygen supplier to the retina. But we can show now that it's an epithelium that modulates the composition of the vitreous. It supplies glucose to the glucose-hungry retinal neurons. It takes up CO2 and it takes up lactic acid. But then the question is, how does glucose and lactic acid being distributed within the retina where there's no retinal neuron or where there's no retinal blood vessels? We have some data on this. I think this is probably the most speculative part of our current data set, but something that we would like to look much more into. But what we wanted to do is try to understand, are there cell types within the retina that could distribute glucose and lactic acid between the retinal neurons? So we did a single cell transcriptomic data set on the zebrafinch retina, where we could see that Müller-Glier cells are extremely high in GLUT1 transporters and MCT1. We could also see that where those proteins are present is primarily in the end feed of the Müller-Glier cells. So in the interface between the retina and the vitreous of the vitreous. So what we suggest could be a mechanism for distributing glucose and lactic acid is that these transporters are present in the end feed of these glial cells. They could take up glucose and release lactic acid and then distribute it into those retinal neurons that needs glucose and needs to get rid of lactic acid, simply driven by glucose and lactic acid gradients between the retinal neurons and the vitreous of the eye. But this is something where we have little functional data, so in case any of you have ideas to how to actually measure if there's a flux of metabolites going through these glial cells, then I'll be happy to talk to you. So this was the mechanistic data, but I'm in an evolutionary group, so we wanted to also test if this is something that is unique to zebrafinches or something that is more widely distributed amongst birds. So we not only did these measurements in zebrafinches, but also in other groups of birds that are more or less closely related to the zebrafinches, where the chickens represent a very early diverging branch of the chickens of the birds. And then we also included crocodiles, turtles, and lizards as the closest related outgroups to the birds. We did the same measurements of oxygen in the eye of these animals, where we could see that the anoxic phenotype is something that is restricted only to the birds and not present in the reptiles, suggesting that it's probably a trait that originated in the crown group of birds after the split from the crocodiles, but before the radiation of the current living birds. We also looked into the evolution and function of the conus and pectin system. So lizards have a similar structure as the conus, or as the pectin, which is called the conus. And we tried to see if they shared the same arterial origin, which could suggest that this is the same structure. And here we could see that they both originate from the ophthalmic artery, at the exact same position of the ophthalmic artery. We did spatial transcriptomics in a multi-species context, where we could see that the conus and the pectin map completely together in a UMAP space, suggesting that they have very similar gene expression profiles, suggesting that these must be the same structures. But when we compare the metabolite transporter expression, so here GLUT1 and MCT1, these are highly expressed in all the birds, but the conus of the lizards do have a little bit of expression of these transporters, but they're not highly expressed compared to the retina. So this suggests that the conus and the pectin represents the same basal structure, but where the pectin then developed a transport capacity to transport metabolites in the early evolution of birds. And then finally, we looked into Müller glial cells function to see if the high expression of GLUT1 and MCT1 within the Müller glial cells is something that is present only in birds or more widely distributed amongst other vertebrate species. So we have the birds up here. They are very high in expression in MCT1 and GLUT1. This is similar patterns to what we see in some other visually dependent animals, for example, the primates. But the expression levels in those species are just much more reduced compared to the birds. So it seems that the birds have just used the same, you could say, building blocks as is present in all the other vertebrates, but have just overexpressed and MCT1 and GLUT1 within the Müller glial cells. So together, these comparative data suggest that in early bird evolution, after the split from the crocodiles, they developed their anoxia tolerance, and this was supported by the pectin being able to provide glucose to the retinal neurons and take up the lactic acid that it produces. And then Müller glial cells in the same phylogenetic interval developed a higher capacity to distribute lactic acid and glucose within the retina. So we suggest that the development of an avascular but very cell-dense retina within birds was supported by this triad of mechanisms that supports the anaerobic ecosystem within the retina. So to summarize, this first part of our bird project have shown that bird retina represents a neural tissue type that functions under the complete and continuous absence of oxygen. It is highly glycolytic, and that is supported by two different lactic acid glucose shuttles within the retina and within the vitreous of the eye of these animals. We show that this is something that originated before the radiation of the current living birds and might already have been present in the dinosaurs. And then finally, we argue that this retinal anoxia tolerance is something that is very beneficial for animals or for birds that fly at high altitude. If we are at high altitude, we increase perfusion of our blood vessels through the entire body to deliver enough oxygen. But this is not possible within the bird retina because they don't have any blood vessels. So if they did not have mechanisms to tolerate the complete lack of oxygen, there was no way that they could use visually guided migration when flying at high altitude. So this is what I had, and I'll be really happy to take any questions.
All right, Salmon. Yeah.
Hey, do you have my voice now?
Yes.
Yes.
Okay, it's really cool topic. I had a couple of questions starting from first. From your oxygen electrode probes, so how you make sure where you place your electrode? So you said like in retina or RP, you have different oxygen pressures. So is it like guided by something or how it goes on?
Yes, ideally we wanted to have done the classic Linsenmeyer electrode measurements where you could both measure the ECG the ERG and the oxygen levels at the same time, whereby you're able to know where do you measure your oxygen levels that you are measuring at the moment. We did not have those electrodes available, so we had to do it blindly, you could say. So we had to use morphological markers of where we were in the retina, and then link that up to retinal morphology after that. So it's not as precise, but it gives the same overall conclusions. So here we use the choroid, so the very high oxygen levels in the choroid, as a marker of now we are in the choroid, and then we map the rest of the profile to retinal thickness or the retinal distribution from histological sections.
All right. And do you think you can do that like by OCT, like while you are doing OCT, you insert your needle and you can clearly see right now where you are.
Yes, you fell out a little bit, but you're right. You could do OCT measurements at the same time if you have like a surgical scope with OCT built into it. And we did that for a few measurements together with some clinicians, where we could validate that what we were measuring under normal circumstances is also what we actually find in the animals.
Right.
But it was not something that we could that we had access to through all the different animals that we studied.
All right. And the other question was that this bird you're studying is like daylight flying bird. Yeah. So we have like some nocturnal birds like fly at night. Do you think that structure would be different in like nocturnal birds compared to the like this bird you studied? Because their retina is different, kind of like as I know. They have like thicker retina because they need more
Salmon,
sensitivity.
it's very hard to hear you right now.
Oh, so what about now? Do you have my voice now? Or?
Yeah, it's better now.
Yeah,
but
I was asking. Oh, yeah.
Yeah, what's your question?
Yeah, my question is this bird you are studying is like daylight flying bird? Yeah,
yes,
like flies in the day. But we have some birds like are nocturnal. They fly at night. They hunt at night, and their retina is kind of different morphologically.
Yeah.
So do you think that structure is different in that bird compared to daylight bird, or?
Yes, so you're right. An owl or an oil bird, they have a rod-dominated retina compared to the cone-dominated retina of most diurnal birds. So there could potentially be other stuff going on there. But what we show is that already in the crown group of birds, all these mechanisms for handling the lack of oxygen were already present. So it means that they have, like, potentially they could have gone back to the, you could say, vertebrate norm, but at the same time they have the molecular machinery to handle everything as the other birds. So I'm not sure. We haven't done any measurements in owls or oil birds, so we don't know.
Right. My last question is that, so since the neurons are kind of far from these structures, as you said, so do you think we have like the same neurovascular coupling function in birds, or it's gone because we don't have vasculature?
I don't think there's a neurovascular coupling because there's no blood vessels. So there can't really be.
So they don't have it, you think?
There's no because there's no blood vessels. But of course, within the pecten, there could be a regulation of blood flow there.
Right. Okay, great. Thank you.
Thanks.
Oh.
Yeah, great talk. I've got a million questions, but I'll try to limit it to just two. I was curious if looking at the glucose transporters in Muller glia, have you looked at Irong Peng's data where they've done sequencing at macular versus, peripheral Muller glia and show that they're different? Is that something that pops up there in the differences since the macula is somewhat avascular?
I have not looked into that, but that's actually a fantastic idea. I'll try to look that paper up.
Yeah, and then a similar question. I've been working with Jim Hurley a little bit to figure out how glucose might move within the Muller glia. Do you think it's just staying as glucose, or do you think it's being processed into something that is then actively moved or helping with a diffusion gradient? Because I can't imagine that a glucose just goes in and comes out the other side. Is there, have you thought about that processing pathway at all?
Yes, because it is long diffusion distances. So ideally, if you could keep a low concentration locally, that would then increase your diffusion rate. This is something that is that some plant cells does, that they actually locally within a cell turns the glucose into like a different isoform that then keeps the gradient going. So this could be something that is going on right here.
And those would then need to be partitioned within the Muller glia. in some way. So that should be something we could find.
Yes, it would.
Okay,
Yeah.
cool. Thanks.
But it's very speculative. But we just see some patterns, and this could be how we could explain that the glucose distribution functions.
Yeah, great.
Oh,
Ian, did you have a question?
yeah, I have a quick question. Sort of curious. I don't normally think about evolution and so forth, but oxygen was, I think, peaked in its concentration in the atmosphere, like 500, 600 million years ago, and it's been decreasing since then, although it's pretty stable now. Just wondering if there's any tracking of the evolutionary traits of the birds and other animals with their oxygen need that paralleled with the changes in oxygen in the atmosphere.
Yeah. Yeah, so there's a lot of evolutionary biologists that always try to link some physiological system to oxygen levels. You could say that all current living animals, their ancestors have at some point been through that transition. So you could kind of come up with that argument for many physiological systems. Well, I don't think that the evolutionary origin of the traits that we see here may have co-evolved with low oxygen levels in the atmosphere.
So just a follow-up, I mean, can you do an experiment on these, the birds in a hyperbaric chamber or something that gives them different oxygen levels? And would that change, anything that you could measure in a way that would be interpretable and interesting?
Yes. Yeah, so one question could be whether the function of the retina is entirely oxygen independent, which we predict it is. We just got our new ERG Celeris system, and the idea for one of the first projects is to try to measure ERG in animals that are under normoxic conditions, hypoxic conditions, or hyperoxic conditions, where the oxygen levels are so high that the retina is in an oxygenated state, to see if they can actually improve vision by having available oxygen in the retina. So that's a project that will be starting up soon. We have done optomotor experiments under normoxia and hypoxia relevant to like 5,000 meters altitude, where we see that they still can track changes in the environment, even though the oxygen levels is only half.
Okay, nice. I say that paper that we essentially did that in vitro, and I'd be interested to see the parallels between the changes in oxygen that we induced and the changes in retinal metabolism versus what you see in vivo.
Yeah, but I can send you that paper.
Yeah, great. Thank you.
Jason?
Two questions. What's the developmental origin of the pectin? Is it, in other words, is the hyaloidal artery and what goes on in mammalian development and with eventual regression, the parallel structure, or is this structure just exists completely separate on the evolutionary tree after, mammals branched off? That's my main question.
To be completely honest, I don't know. I haven't looked into the development of the pectin, but it's So I honestly don't know, but it's something that would be super interesting to look into.
It's just always, it links to the question, which also is unanswerable, but obviously primates have foveation, birds have foveation. They were separate evolutionary events, but they both, but presumably the things that led to the process, there was the same pressures, and a thick retina I could see as being a pressure, for foveation. But in mammals, our retinas are not as thick as birds. So I'm trying to sort of link the evolutionary logic together. But anyhow.
Yeah. Yeah, so it has probably been like selective pressure on having high acuity vision, which you can do by having foveation, because you like increase the photoreceptor density locally within your retina. So I guess it, as you also say, that it has just been the same kind of pressures for having high visual acuity that have led to the same morphological phenotype in both birds and in mammals.
Okay, so you have people complimenting the talk in the chat. So clearly well received. I have a couple quick questions, I think. The first is just kind of like technical. You had these glycolysis scores, oxidative scores. Is this just like how did you arrive at this score?
So for each of our gene expression spots, we basically had a whole transcriptome. So where we had measured like all the different genes that are being expressed under that spot. Then I calculated what is the proportion of genes that are within the glycolytic pathway, and normalize that to the average expression of all the genes, of all the other genes within that gene expression spot. So I don't think the numerical value provides much meaning, but what is good is that you can compare like expression scores across your entire retinal morphology and see where is specific pathways upregulated compared to other parts of the tissue.
Yeah.
So it's like the relative upregulation of a specific pathway compared to the average gene expression within that spot.
Do you have any sort of, especially for things like the OXPHOS score, any kinds of electron microscopic images that would really support the scoring metric by showing mitochondrial density or anything like that really aligns well with the score?
Yeah, yes. So we have done mitotracker experiments, so where we injected mitotracker into anesthetized animals and then did microscopy afterwards, and it corresponds very nicely with the values that we get here, with a very thick mitochondrial area within the photoreceptor outer segment, and then loosely distributed mitochondria through the rest of the retina.
Outer segment or inner segment?
In the outer segment, so like similar to mammals.
Most of the mitochondria in mammals are in the inner segment, but
yes. So in the inner segment, yeah.
And then, so my other question, this is kind of maybe along the lines of the questions other people have been asking. Do you have, so of the different birds you've studied, do they have heights that they generally prefer to fly at that are sort of clearly stratified? And if so, does that relate at all to pectin size?
So the animals that I study, so the chicken is of course down to like walking on the ground, and the zebra finches and pigeons, they are flying at normal altitudes. We would have liked to get our hands on some of the high flying birds to see if they potentially have a more elaborate pectin that could support more glucose into the retinal neurons when flying at those altitudes. I don't think people have studied that particular aspect of pectin function, but that's of course relevant now as we show our data. But people have looked in, like compared birds of prey, where birds of prey that have exceptionally good vision, so an eagle or a hawk, they have more folds within the pectin, so a higher surface area, which makes sense because they probably also need more glucose in order to feed their retinas.
But doesn't the pectin interfere with vision? Aren't there like floaties in the eye?
Yes, it must do, because it is a really big structure. When you see like when you open an eye, it takes up a lot of space. So they must have a huge like slit of a blind spot within the retina.
Yeah.
But if they're flying, Jim, and it's moving around, maybe the blind spot is changing enough that you kind of don't have to deal with it too much.
Yeah, I can't argue with it. It works. Birds are they thrive. Yeah. So
Yeah.
All right. Does anybody have any other questions? Kieran.
Hi, great talk, Christian. I have a small question regarding the intense activity in birds. So, as you mentioned that the birds they consume glucose at a much higher rate. So, and that all experiments I think were done at stationary level. So, in under intense activity conditions, like when they are focused on a prey or when their retina is like super active, in those conditions, do you think that the glucose consumption is happening at like way more higher rate? Is that what you are saying?
Yes, we didn't measure it, but there's I mentioned that preprint that got published or that got released a few months ago, and they show that under conditions where the animal is more visually active, they use the proxy of when they're together with other pigeons. Then they are more visually active, and their glucose consumption within the retina is increased. So indeed they modulate the glucose demands depending on their visual needs.
Oh, okay. Yeah. Thank you. Great talk.
Thanks.
Okay. Thank you everybody for coming. I'll stop the recording.